Okay, so we've gone through QC, some visualization. The next step, having sort of visualized our data with PCA or TISNE that can highlight some aspects of variation between our population of QC'd cells. What we want to do next is try to identify what could be confounding factors in our analysis. And so that's what we'll cover in this chapter now. So there are a large number of potential confounders, artifacts and biases that can be introduced into single-cell RNA-seq data, and that goes from the very first aspects of sort of the data production process, from isolating cells through to sequencing and all the rest. And part of the issue we have, or a challenge we have to face, is that it's very difficult to carry out a true technical replicate in single-cell RNA-seq data. So in the bulk setting, we can take an RNA library, a pool, and we can take different aliquots of that, process them, sequence them differently, sequence them, and look at actually quantify, directly measure the technical variability between what is produced, the variability between two libraries that started from the actual same RNA source. And we can then contrast that with the degree of variability we see if we take, say, two different samples from the same mouse or the same patient or the same cell line, where there's actual biological variability between the RNA samples that we're starting with. And so in bulk, it's quite, we're able to do that quite successfully, like try to firstly measure degrees of technical and biological variability, and then develop models or utilize models that are also able to distinguish technical from biological variability and use these in analyses. So why can't we do the same in single cells? The problem, of course, is that as soon as we assay a single cell, it's gone forever. And part of the motivation for doing single cells is that we don't think that cells, even under nominally the same conditions, are completely homogeneous. If we did believe that, then we may as well just do bulk sequencing and save ourselves the trouble of everything we've discussed today. So a challenge for developing methods and for also doing our analyses is that we can't, it's difficult to completely separate technical sources of variability from biological sources of variability that we're interested in when we have a set of cells. Having said that, there are certain things we can expect. So batch effects are an aspect that we have to face across all aspects of genomics and beyond. It was true of microarrays, that if you processed the same samples on different microarrays on different days with different ozone levels and all the rest, then you would see batch effects evident in the measured results. The same was true for bulk RNA-seq samples, so processed at different times by different people in different labs. The measurements made are different, and we can notice that. The same is also true for single cells. So depending on the plate that your cells appear on, or depending on when they were processed or who did the processing and all the rest, we can see large-scale differences in measured expression levels that we can really sort of pin down to some of these technical or experimental artifacts that we're not interested and would ideally like to remove as sources of variation from the data before we try to answer biological questions with the data. So things like PCA and so on are quite useful for visualizing the cells, trying to find out structure, but there are a few more tools in SCADA that we can apply to get sort of deeper into identifying confounding factors. And so here again, we're back to the UMI data, and so the UMI QC data set is what we'll address here. So having QC'd and filtered out the cells that we don't want to use, now we can sort of think about what processes, or especially technical sources of variability, seem to be underlying variation between our cells. So if we look at that PCA plot again of the QC data, this is what we see. So we see up here a group of cells from replicates from a single patient, clustering visually differently from the others. And what we can do is, with a particular plot in SCADA, is say, okay, we see that component 1 here explains 17% of the variance, component 2 explains 7% of the variance, subsequent principal components will explain correspondingly fewer, lower proportions of the variance. But to interpret what the PCA is picking up, we'd like to know what variables seem to be driving this signal in the PCA. And so with the plot QC function and the type equals find PCs, we can take a variable of interest, so in this case total features, so the total number of detected genes per cell, and we can find which PCs are most correlated with that particular variable of interest. So we're starting with total features. We say, okay, what principal components are most correlated with total features? And here we see that, which is very typical for single cell data, that in this panel of the plot here, component 1, so principal component 1, is very strongly correlated with the number of genes detected per cell. And so we can sort of look at that and say, okay, it looks like the major source of variability is associated with the number of genes detected in the cell. And this is very typical across experiments. And so we can also plug in any variable we like, so any of the QC metrics that have been computed and added to our phenodata table, we can look at there. If there are sort of biological groups that we know about or other annotations for the cells, we can also look at how they correlate with the principal components. And this plot works whether the input variable is continuous, like total features here, or if it's categorical, like groups, group 1, group 2. So if it's a continuous variable, you see a plot like this, a normal scatter plot with a fitted curve. If it's categorical, then you'll see like violin plots for the two groups. So that regardless of what the type of the variable, you'll get a sensible plot out that will show which principal components have the highest R-squared value for that particular variable of interest. So we can also switch to thinking about what explains variability at the gene level. And so also with the plot QC function, we can produce a plot with this command for a set of variables of interest, so like total features and things like that. And what we do to try and understand what's sort of explaining variability at the level of individual genes is that we can fit a linear model for a variable of interest to a gene's expression level values and compute the marginal R-squared. So from that linear model, how much of the variation in expression for this gene is accounted for by this particular variable of interest. So we can do that for total features, for example. And so for each gene, fit a model where we regress the expression values for the gene on this total features variable, and then find out through R-squared, use that to define how much variance is explained. And when we do that for each gene, then we have 10,000, 8,000, however many genes, we have that many R-squared values for that particular variable of interest. So that gives us a distribution of R-squared values for each variable of interest. And that we can use to get an idea of like sort of, on average or across all genes, which variables seem to account for a lot of variation in the gene expression values. And so doing that, we can get to a plot like this. So here in this plot QC command, we're saying, okay, we want to look specifically at total features, total counts, batch, which we know about, the individual, which we know about, the percentage of counts that come from ERCC genes, and the percentage of counts that come from mitochondrial genes. And so we can see here that each of these variables appears as a curve, a density plot here. And so on the x-axis, we have the percentage of variance explained, so R-squared, for each of these variables. So we're showing the distribution of R-squared values for all of these variables. And so you can see this dotted line is at 1%, so we're sort of interested in what's happening up this end, so where, what variables, for example, explain more than 10% of the gene expression variance for a large proportion of genes. And so density curves that have a sort of a mode and skewed towards the right-hand side, that's saying that this variable marginally explains a lot of variation for a lot of genes. Distributions that are skewed to the left or have a mode towards the left explain very little variation for a lot of genes. So when we look at batch, for example, we have this big peak here that's centered around just under sort of 10%. So here we could say, most genes have, say, between 5 and 10% of their variation could be explained by batch. On the other hand, so the mode for total counts or total features is above 10%. So most genes, or a majority of genes, probably have more than 10% of their variation explained by total features or total counts per cell. Then for something like the percentage of counts coming from mitochondrial genes, that's much lower. So that mode centered at less than 1%. Yes?
What's the expected distribution for just noise?
It's a good question. It's a good exercise for you. Fit a Gaussian variable.
Do you have a noise model for gene expression data that works?
What do you mean? What do you mean a noise model? I mean,
what's the typical model of the noise in gene expression that you would use for simulation?
well, so bulk counts are fairly reasonably log normal distributed. So once you take the log of so these normalized counts per million, which is what we're using here, you get reasonably close to a normal distribution apart from the excess zeros in single cell data. So this is fairly unsophisticated, and so that's why I wouldn't necessarily use this model for, say, differential expression testing or something like that. You'd want something more closely tailored. But for a broad scale look at sources of variability in the data, I think fitting a linear model, so a normal linear model, to the log 2 of the normalized counts per million, I think is a defensible thing to do. But you may disagree. But in this case, yeah, I mean, well, if you wanted to have a look at like the effects of, say, it's completely uncorrelated values, then you sample from a normal distribution, fit that as a variable, and you can see what it looks like. Yeah, so in this particular case, I guess this highlights something similar to what the correlation with the principal components did, which is like total features or total counts are really important sources of technical variability that affect our comparison of the different cells. So here we have a known batch effect that was sort of introduced by design in the experiment so that we can actually investigate it. Total features, total counts really come up as important confounders or sources of technical variability, and that's common across most single-cell data sets. There, of course, are other factors that one might want to account for, and some of these require external or extrinsic information. One thing that we might consider in many single-cell experiments is the effect of cell cycle on the measured expression levels. And so in bulk samples, the general assumption is that if you're averaging over 10,000 cells, they may be in different phases of the cell cycle at the point that you assay them, but this sort of averages out, or comes out in the wash, is the general idea. When we're looking specifically at single cells and are interested in the differences between single cells, then if this particular cell is in phase G1 and this one's in S phase, then we might be finding many differences in gene expression, or the cells might like look quite different at the transcriptomic level due to this latent factor of the cell cycle rather than the biological process of interest, which might be differentiation or something like that. So there are now a couple of methods out there that try to infer cell cycle phase from the gene expression values. And so SC-LVM, which is mentioned here, is one. There's another method called cyclone in the SCRAN package, which also does this. And so we can use statistical approaches to infer cell cycle and then fit this as a confounder or a covariate in models that we're interested in, or try to remove them by regressing them out in one way or another. As well as that, so that those ones are specific methods for cell cycle. You could also imagine other underlying processes affecting the transcriptome that we measure, like apoptosis or other biological pathways or processes that may not be of interest. And so in general, there's a whole sort of area that tries to discover these hidden or latent factors, tries to identify things that could have large-scale effects on the gene expression measurements that we make, so that they can be included or modeled explicitly or sort of regressed out in adjusted or corrected data that we put into downstream models. So when it comes to thinking about confounders, as well as the very technical things like number of genes expressed, the total read counts per cell, and batch, which hopefully you know about from the experimental design, then there are things that we can't necessarily control, like generally cell cycle or other things, which you might want to take into account in analysis depending on exactly what you want to do. And so worth thinking about or bearing in mind that these sort of latent variable or hidden factor models exist and could be applied in many appropriate scenarios in single cell data. As well as all that, we have differences in protocols when it comes to coverage of each transcript, as we've discussed, so full-length transcripts or 3' bias or just a tag from the end, biases that can be affected by GC or AT content, and all that sort of things. Some of them are sort of beyond the scope of what's possible with sort of analysis methods at this point. Some things are only going to be solved by the people who develop these technologies doing a better job. And in the meantime, we sort of do the best with what we can, given the biases that are inherent in the technologies that we have available. The other things we try to model, or at least detect and be aware of before we draw strong conclusions from our data. So in there, we covered sort of looking at correlations with PCs, looking at explanatory variables and other confounders. That was all on the UMI data, so over to you for this exercise now. So try out the same things with the read counts of the bleach-shape data. So that's the next topic, and we'll give you a few minutes to work through that and then discuss. Any questions now before we continue on with that? Yep.
Will you be covering some of these latent variable models later on in the course?
So the question is, are we covering latent variable models later in this course? I think the answer is no. This is like still all of the methods in single cell analyses are developing, and some areas are more mature than others. I'd say the latent variable modeling is still at the more experimental end of the methods development. I think there are some things that I know about that are in the works that I think will prove very useful. I think the best established ones at the moment are this SC-LVM, so which is like a latent variable model specifically, which really digs into the cell cycle. There's the cyclone method from the SCRAN package also for cell cycle. And then I know the Pagoda package also has some approaches to trying to identify biological processes generally speaking, so going beyond cell cycle, that are active or sort of prominent sources of variation in your data, that's not a sort of a formal latent variable model, although I know some people are developing sort of formal latent variable models to address these things. Okay, so if we apply those same ideas to the read data, so we had a similar-ish looking PCA plot. If we do that approach of finding PCs correlated with total features, again, principal component 1 is strongly correlated with that. Correlation is slightly lower than it was for the UMI data, but still it's clear that total features is the main driver of variation as picked up by PC1. Here, interestingly, for the UMI data, we saw total features and total counts seem to be sort of explaining more data, or more, if we compare with this one, total features, total counts explained a bit more variation for more genes than when we look at the same plot in the read data, possibly, again, reflecting just the greater noise levels in the read data, so none of these particular variables explain as much here. Batch seems to be a slightly stronger effect than it was previously, but fairly marginal, I suppose, there. Okay, so that is overview of some approaches to identifying confounders, and the next step we're going to go into in the next chapter is normalization for library size. So Tallulah will do that in a minute. Any other questions coming to mind before we approach normalization? Yep.
Might be a very silly question. So all the log2 transformations which were done before were done on the raw counts, right? No, in log2 it was.
Right. So
When you use the log2 and when you use the other ones, which are going to like the PKM and the other, why don't we use that directly instead of?
So the question is, when are we using counts? When are we using just like log counts? And when are we using something? Yeah, so I guess we glossed over this at the start, but when you build an SCE set object with the new SCE set function and give it counts, then the counts matrix will be retained in the object. But what appears in the expression slot, so the EXPRS slot when you do that, is the log2 of the counts per million, so yep, plus an offset, and by default that offset is 1. So we sort of touched on that at the start. So in that respect, when you look at the EXPRS matrix, the zeros in that matrix correspond to 0 counts, but the actual other values that we're getting are these log2 counts per million.
These are just for library size?
Yes, so this is just counts per million, so coming directly from the library size, that being the total counts observed for the cell. So there are more subtle ways of doing that library size adjustment, and that's what we'll discuss in this normalization, throughout these normalization approaches. So for sort of just visualization and discussion purposes, we were showing what it looks like, if you say do PCA on log2 counts without this normalization of library size between cells, but by default in the package, we do that because in general, there are always, and this is true in bulk as well, always differences in sequencing depth and therefore library size between samples. So to make them at all comparable, we need to account for that. And so by default, the SCADA package does that if you provide it with a count matrix when you build the object. And so these PCA plots, for example, and these QC plots, then the expression values that we're thinking about in here are log2 counts per million with that small offset. Yep.
So I think the log counts that we created earlier was log2 of raw counts.
Yep.
So that would look like there just the log of
So we defined a specific slot in the object, which was log2 counts, which was log2 raw counts. That's distinct from what's in the EXPRS slot. And so what's produced in the EXPRS slot when you create the object is these log2 of the counts per million. Right, so if we specify, right, okay, here, yeah, fair enough, yes. So here we've specified that we want to use log2 of the raw counts as expression values, so that's exactly what it does. I guess in the, no, so the default, so there's counts, there's log2 counts. Not sure if I have an example. If you don't specify this argument, the default is EXPRS. So the default, if you don't specify that argument, will be log2 counts per million, which is generally a much more appropriate thing to use than raw counts or log2 of raw counts. Yeah, good to clarify that. Yeah. Okay, we're going to cover CPM in the normalization chapter, so hopefully things that are not clear yet should become clear as Tallulah explains. So I think, indeed, yeah, this is sort of part of the discussion of building up from like raw counts to normalized values downstream. We're sort of presenting this as something, well, hopefully explaining what we're doing, but Tallulah's going to explain now more about the normalization and what we do. So.
Okay, so now we're moving on to the normalization section. So as you saw in the previous chapter, the total number of features or the total number of counts you have in each cell is one of the biggest differences you'll see between the two cells you have, any two cells, and it's one of the biggest confounding factors. So to deal with this is usually called library size normalization because the total number of reads per cell is referred to the library size. And they differ a lot because you're sequencing this in a highly multiplexed form, and just by random chance, different cells will get different amounts of coverage. And also during the library preparation steps, some library preparation on some cells may have been more efficient than on other cells, leading to slightly different library sizes, which are then sequenced to different depths at the end. So some quantification methods, such as cufflinks and RSEM, actually incorporate library size when they estimate the expression level for your genes in their model, so then you don't need to do library size normalization after the fact. But typically you'll be producing raw read counts or raw UMI counts, in which case you need to normalize for library size. And this is typically done by calculating some sort of normalization factor in various different ways as an estimate of the library size for that particular cell, and then dividing every expression value in that column, so for that cell, by that normalization factor. And there are lots of methods that have been developed for bulk RNA-seq, because you get the same problem for bulk RNA-seq, that can be applied to single-cell RNA-seq, but we're going to argue that you probably should not be doing that. So the simplest one is counts per million, which you've been just talking about a bit already, where we basically take the total number of molecules you have for each cell, divide the column by that, and then multiply by 1 million to get the counts per million. This works quite well. However, you can have a problem if you've got some genes that are both highly expressed and differentially expressed across your cells. In this case, the total counts can be determined in part by the differential expression of those highly expressed genes, which can then cause this normalization to fail and will actually lead the more lowly expressed genes to look like they're differentially expressed when they're actually not, because of the normalization. To deal with this, there have been lots of variants proposed. So one of the most popular one is size factors, which is from DE-seq, which essentially uses the geometric mean of the expression of each gene across all cells, and then using the median value of the ratio of the gene expression in your cell, in a particular cell, to this geometric mean to estimate the size factor. The problem with this is that if you have a 0 for a gene, for the gene expression level in any cell, then the geometric mean for that gene across all cells will be 0, and you will not be able to use that gene in this normalization factor. For bulk RNA-seq, that's not really a problem because you don't have a lot of zeros. For single-cell RNA-seq, this can mean you're using 5 or 10% of all of the genes in your data set in your normalization, which can, again, lead to biases in your results. Yes, this is how you would calculate it. And later on, we're going to be calculating this using Skater, where it's called RLE for relative log expression, but it's essentially doing the same thing. It's not quite the same, I don't think, but it's almost the same. Another method is the upper quartile method. Basically, you calculate the 75th quantile, so the upper three quarters of the counts in each cell, and then normalize using that as the denominator. Again, this works very well in bulk RNA-seq methods, but if you have very shallow sequencing, particularly in, say, a drop-seq type experiment, then the upper 75% quantile in some of your cells will be a 0, which obviously doesn't work because dividing by 0 will give you an error. So you may, some people will just take the 75th quantile of the non-zero values in each cell, which can improve this, or you can use a higher quantile, such as the 99th quantile, which is the default in Skater, to avoid this problem of sometimes getting a 0 for your normalization factor. And this is how you calculate it. So apply to each column. Apply to each column of this matrix, this upper quantile. We calculate the quantile. And then normalize this to the median of the UQ so that you're not shrinking all your numbers, your expression values too small. And then divide the entire matrix by that normalization. Yet another variant on normalization factors is called TMM, which is the weighted trimmed mean of M values, which basically takes the expression value in one particular cell and compares it to your reference cell, which is picked arbitrarily. And then it looks at the log 2 fold changes of the expression value in your cell of interest compared to this reference. And then those become the M values, and then it calculates the mean of that. First it removes the extremes of that, and then it calculates, weights them, and calculates the mean of those to give you a normalization factor. And then again, you just divide each cell by that normalization factor. So again, because you're trimming the values, you're leaving off the top 30% in most cases. If you have, again, very shallow sequencing of your single cells, you can end up with having zeros as your normalization factors using TMM occasionally. So far, the only good single-cell specific normalization method developed is this SCRAN package, which is a variant on CPM, where, again, you're going to be calculating a normalization factor for each cell, but to deal with the problem of having a large number of zeros, it pools cells together first, calculates the normalization factor for each pool, and then then deconvolutes a normalization factor for each cell from a whole bunch of different pools using different combinations of cells. And then as we've been talking about before, we're going to be looking at the PCA plots to see how well these normalizations are working, as well as calculating using this function, the calc RLE, which will calculate the relative expression in one cell versus the other cells. So it's basically taking the median expression of a gene across all of the cells, taking the expression in this particular cell, dividing it by that, and then just log transforming that. So if one particular cell, and then you will take the median of that across all of the genes for that cell. If your cells are well normalized, you'll have about 50% with that will be above the median and 50% below the median. So the median across all of them will be close to 0. Whereas if you have badly normalized cells, then you'll have a surplus of gene expression values that are above the median, which will give you a positive, or a surplus of genes below the median, which will give you a negative RLE score. So Skater very helpfully has this wrapper called calc norm factors, which enables you to calculate all of these different normalizations very efficiently on your data set, just by specifying a different parameter. And we're going to continue using the same data set as before. So if you load the data up, we first have the PCA plot of the raw counts, which I believe you've seen before, where we have one patient separated and the others less so. Looking at just the log 2 counts. And then if we calculate the RLE on the unnormalized data, you can see that we've got quite a few cells that have positive or negative scores. They're not all close to 0. And then, so CPM is already calculated by default and put into the expers slot in Skater. So into the PCA of that, and you can see that now all of the patients are quite separated from each other, although you can also see some batch effects affecting this data. And if we look at the RLE, you can see that they're all essentially 0, indicating the data is now well normalized. And if we use TMM, so here we can change the method to TMM to calculate our expression values. And then we save this in a slot called norm counts and plot the PCA. You can see it's not as good as the counts per million were. But you can still see that each patient is sort of separating out individually, but there's still a lot of noise in there. And if you look at the RLE, again, there's lots of cells that have non-zero RLE values, showing that this method is not working as well as counts per million. And then we can perform scran. So here first it clusters the cells into clusters with a minimum size of 30. It then pools cells within each cluster. Here we've set the size to 15, so it'll randomly select 15 cells from that cluster of 30 or more to form a pool, and it'll form many, many pools for each cluster. And then it will compute the size factors for each of those pools before deconvolving them back into size factors for each individual cell. And then we apply the normalization using that function, and the normalized values will get stored into the expers slot of Skater. So we can plot the PCA, and we see that it looks essentially the same as counts per million for this data set. And this is because these cells are quite uniform in size, and there's not a lot of differential expression going on. Whereas if you have cells where you have very large differences in differential expression across the cells, or cells of different sizes, you'll find that scran will work much better than counts per million. And if we look at the RLE, again, they're all very, very close to 0. Then we have size factors. This is from DSeq, which gives this, which is not as clear. Again, we've got overlap between, quite a lot of overlap between the patients there. And we've got some more larger RLE values, although not as bad as TMM. And upper quantile, which looks very similar to RLE. Although using the RLE statistic, it looks quite a bit worse. So any questions on those methods? Okay, the next method is different from the above. Now we're not going to be dividing by a normalization factor. Instead, we calculate the library size of each cell, find the smallest one, and then downsample all of the other cells randomly so that the total counts are the same as that smallest library. The benefits of this is that it will eliminate biases due to different numbers of genes detected. Because before, when we're just dividing, if you have a non-zero, after you divide by some normalization factor, you'll still have a non-zero value. Whereas if you do have a 0, you'll still have a 0. Whereas downsampling, some of those non-zero values will be turned into zeros appropriately after you downsample. The drawback is that it's not deterministic. So if you downsample multiple different times, you'll get slightly different expression matrices at the end, which can be important if you're looking at rather small differences and trying to do very fine analyses on your data. So this is a function for doing this. So first we calculate the minimum library size, and we find the relative size of this library compared to the minimum library size. And then here we're using the binomial distribution to randomly downsample. And then if we plot the PCA of this, you can see again, very nice differentiation of the different individuals, as well as a little bit of batch effect, but not as bad. It looks pretty close to the counts per million case. And if we look at the RLE, it's now completely flat because all of the library sizes are now exactly the same. So the final topic is to normalize for gene or transcript length, because if you have a long transcript, you have the potential to have many more fragments come from that one transcript than if you have a short transcript. And this can lead to long genes having higher expression values. This has been noted a lot in bulk RNA-seq, that long genes have higher expression values on average in bulk RNA-seq than short transcripts. However, this is not so much the case in single-cell RNA-seq, because in most cases, as we saw earlier, you're not sequencing the entire transcript. You generally have a very 3' biased coverage of your transcript. So often, normalizing for the full gene length or full transcript length will actually introduce new biases into your data because you haven't actually sequenced that whole length of the transcript. So sort of use at your own risk, check how biased your data is towards 3' end before applying these. So here, I believe we're going to check here if we have a correlation between gene length and expression level. So first we have to get the gene lengths from Ensemble, and that's how you do that using Skater, which is much simpler than having to do it by yourself. From Biomart. Again, this is Biomart, so make sure you're using the right IDs for your data sets. Some data sets will have different gene IDs. And we have to filter out those genes that we don't have the correct annotations for. And calculate the length of that of each gene. And then we can plot the expression versus the gene length. In this case, because this is UMI tag data, we're only sequencing the 3' end. So we don't expect there to be any correlation between gene length and expression value, and we don't see one. So in this case, there would be no need to normalize by gene length. So we shouldn't be using FPKMs or TPMs for this data set. But here's how you would do it anyway. In Skater, so Skater will automatically do this for you once you've got the known gene lengths. And this is what you get afterwards, which isn't very helpful, but we've already discussed why that's not helpful for this data set. And for you to work on now is to repeat all the steps with the reads rather than the UMI counts. Maybe. If you're using, so the question is, if you're using full-length protocol, should you be normalizing by gene length? Yeah, in bulk RNA, definitely you should be. But if you're doing full-length single cell, full-length single cell often isn't really full-length. Often, it's been shown that the reverse transcription is not as efficient, and you're only getting up to a maximum of sort of 3 kilobases of each transcript. So genes that are bigger than 3 kilobases long, you'll actually be over-normalizing if you apply these methods. There's yes. Where was that? This one? Yeah, so here, yeah, so this was supposed to be full length. Yeah. This was supposed to be full length sequencing, but you can see that it's really not. So in this case, we tried normalizing for gene length, and actually we ended up over-normalizing, and the very long genes had really, really low expression. So, yes, if you're using UMIs, you never should be doing this at all. If you're using full length, then you should really look at your coverage first. So I know that because I've developed a method that basically looks at the relationship between the dropout rate, the dropout rate, so the number of zeros, and the mean expression. And there's a very close relationship between those. So if you're over-normalizing, you'll find that the long genes are all shifted to the left, so they have low gene expression relative to the number of dropouts. Soon. There was a paper published a few months ago that has developed a method to do that, but they haven't created a R package yet. Yes. Yeah. So imminently coming. I keep emailing them to see if it's out yet. I haven't checked that one yet because it's not available, but it looks like it will be better. It doesn't have a name yet. Yes, it's not a package, it doesn't have a name. I can't remember. I will post it on the course website tonight. Or maybe maybe in a week it'll be on the course website.
This is maybe a point you made about like gene length sort of adjustments. Is it where appropriate that could be useful for anything where you're comparing expression values across genes, but they're not necessarily always appropriate for doing like differential expression testing.
Yeah, for differential expression, it doesn't matter because you're comparing the gene to itself. It's mostly if you're looking for maybe marker genes, if you're looking for which genes are most highly expressed in your cells, then it will matter. Or potentially gene-gene correlations might be affected. But if you're just looking at differential expression of a gene in different conditions, it doesn't matter. Yeah. Yeah. The bulk RNA-seq packages will normalize them for you. Single-cell DE packages don't necessarily normalize them for you.
Strictly speaking, like DEC2 and EDGAR use the raw count, but make adjustments in the model by way of offsets to account for differences in library size.
I don't know what
Yeah, no, that's a valid question.
Yeah.
What we found from work on the differential expression methods for bulk was that getting the relationship between the mean variance, characterizing the mean variance relationship was the most important thing when it came to getting good differential expression results. And I think that's still an open question in single cell area. What is the right model for addressing this?
Yes, tomorrow. Yeah. Mm-hmm. Potentially, yes. So, sorry, the question is whether you can downsample multiple times and then average across them. Potentially, you could do that, although you may increase the number of non-zeros again by doing that. So you'll probably just end up getting something almost the same as counts per million in that case. And so it will always be downsampled, but the main benefit of downsampling is that you have the appropriate number of zeros in each cell, which you wouldn't have when you're averaging, because you'll have the zeros will be in different places in different downsamples. Okay, so now we're going to take up the normalization of reads exercise. So again, we have the raw PCA plot, which doesn't look particularly good, and again, fairly wide RLE distribution. Now doing CPM normalization, the RLE looks a lot better, but the PCA still doesn't look terribly good for the reads. TMM, the PCA looks a bit better, though it's hard to tell really, and the RLE looks quite awful. It's not helping at all. I think it's making it worse. For I think this is Scran method, looks very similar to CPMs again. because our data set is quite homogeneous. Size factors looks quite similar to TMM once again, as does UQ, which don't work so well on the counts. And downsampling. So we've also done some downsampling on this one, and here the downsampling doesn't seem to have corrected very much for this data set. For the reads, and then we did FPKMs and TPMs again. But again, this isn't relevant because we're only sequencing the 3' end of the transcripts, not the whole transcript. So it doesn't really make very much difference to the PCA plot. So now I believe I'm handing it back to Davis.